All right, let's dive right in. Today, we're talking about a massive shift. I mean, a truly seismic shift in how we fight cancer. We're moving away from the old brute force methods and into an era of intelligent, precision guided treatments. So, how does this new arsenal actually work? Let's break it down. Okay, so to really get what's so revolutionary here, we've got to look at the old guard versus the new. And this isn't just a small improvement. We're talking about a completely different playbook for how we think about what a cancer drug should do. And this slide lays it all out perfectly. On the left, you've got traditional chemotherapy. Think of it like a cannon. It just blasts any cell that divides quickly. Sure, that includes cancer, but it also hits healthy cells in your hair, your stomach, which is why we see all those brutal side effects. It's cytotoxic. Its job is to kill cells. But on the right, you have targeted therapy. This is more like a guided missile. It's engineered to hit a very specific target that's mostly found in cancer cells, leaving the healthy ones alone. And often these drugs are cytostatic, meaning they just stop the cancer cells from growing. Huge difference. So, if we're talking about guided missiles, what are they exactly? Well, today we're going to explore 3 key strategies in this new arsenal. Each one is designed to attack a very specific vulnerability that cancer cells have. First up, we're going to see how we can basically starve certain cancers by cutting off their hormone supply. Then we'll get into the biggest class of these drugs, the ones that work by jamming the internal communication signals that are screaming at cancer cells to grow. And finally, we'll look at a really clever strategy that actually uses a cancer cell's own fatal flaw against it. All right, let's start with endocrine therapy. See, some cancers, especially certain types of breast cancer, use hormones like estrogen as fuel. They literally feed on it to grow. So the approach is pretty logical, right? You just cut the fuel line. One of the most classic ways to do this is with a class of drugs called SERMs. And the most famous one, by far, is tamoxifen. So how does it work? Well, it binds to the estrogen receptor inside a breast cancer cell. Think of it like this. It jams the lock so the estrogen key can't get in to turn on that grow and divide signal. The cancer's proliferation just slows right down, making it a cytostatic drug. Now that word selective in the name, it's super important, and this slide shows you why. In breast tissue, like you see on the left, tamoxifen is a blocker. It does exactly what we want. But in other tissues, like the uterus on the right, it can actually act as an activator. This kind of two-faced nature is why it can have some serious side effects, like raising the risk for uterine cancer. It's a powerful tool for sure, but that selective part isn't always perfect. But hey, blocking the receptor? That's not the only trick up our sleeve for cutting the fuel line. A different class of drugs called aromatase inhibitors, they just go straight to the source. They work by shutting down a key enzyme that prevents the body from making estrogen in the first place. Simple. No estrogen production, no fuel for the tumor. Okay, on to our next strategy, and this one is a huge deal. It's the largest and, you could argue, most revolutionary class, kinase inhibitors. So inside our cells, we have these proteins called kinases. They're like little switches relaying signals. The problem in many cancers is that one of these switches gets stuck in the on position, and it's just constantly screaming, grow, divide. These drugs are designed to find that one specific broken switch and shut it off. And really, the poster child for this whole idea is a drug called imatinib. You might know it as Gleevec. This one drug completely changed the game for a disease called chronic myelogenous leukemia, or CML. I mean, it turned it from what was basically a death sentence into a manageable chronic condition. It was one of the first true home runs for targeted therapy. So what exactly was the problem that imatinib was built to solve? Well, in CML, this really bizarre genetic accident happens. As you can see here, pieces of 2 different chromosomes literally break off and swap places. This mashup creates a totally new mutant protein called BCR-ABL. And that is the broken kinase switch we were talking about, a switch that is just stuck on 24/7, driving the cancer forward. And here, this is the absolutely brilliant solution. Scientists designed the imatinib with a very specific molecular shape that fits perfectly into a little pocket on that faulty BCR-ABL protein. It's like a custom-made key for a very specific lock. By plugging that hole, it physically blocks the protein from sending out its nonstop growth signal. The cancer cell stops dividing, and the disease goes into remission. It's just such an elegant, precise solution. Now, that BCR-ABL protein isn't the only broken switch out there we can target. Not by a long shot. Another huge one is a receptor on the cell surface called HER2. In some breast cancers, the cells just go nuts and make way too many copies of this one receptor, which creates a whole different kind of always-on signal. And you can see the difference so clearly here. On the left, a normal cell, just a few HER2 receptors. But on the right, you have a HER2-positive cancer cell. It is just covered in them. They act like thousands of tiny amplifiers, picking up even the faintest whisper of a growth signal and just blasting it at full volume inside the cell. And of course, there are targeted drugs designed to go in and shut down that massive overamplification. And this whole idea of blocking signals, it goes even further. We have drugs that block a signal called VEGF, which tumors use to trick the body into growing new blood vessels to feed them. We have others that inhibit a central control protein called mTOR. In every single case, the strategy is the same. Find a critical communication line the cancer depends on and cut it. All right, our third and final strategy. And honestly, this one is just so clever. Instead of just blocking a growth signal, these drugs actually exploit a weakness that's already in the cancer cell. They turn the cell's own broken machinery against it. So here's the genius of it. Healthy cells have a couple of different ways to repair their DNA. Now, some cancers, especially those with a BRCA mutation, have already lost one of those repair pathways. They're kind of limping along, relying on this one backup system that uses a protein called PARP. So what do PARP inhibitors do? They come in and cut that last rope. They block the backup system. And with no way left at all to repair its DNA, the cancer cell just builds up too many errors and self-destructs. It's a kill switch that only works because the cell was already defective. So, when you take a step back, what does all of this really mean? It means we're moving away from treating lung cancer or breast cancer as one thing. We're moving toward treating a cancer based on its specific molecular driver. Is it a BCR-ABL mutation? Is it HER2 amplification? Is it a BRCA defect? It means the future of cancer therapy is incredibly personal. If you take away one single thing from all of this, let it be this. This isn't just about making new drugs. It's a whole new philosophy. It's about doing the molecular detective work to find out what specific engine is driving a person's cancer and then picking a drug that was designed specifically to shut that one engine down. And this revolution is absolutely still unfolding. Every new pathway we discover, every new vulnerability we map out, it just opens the door for a new targeted drug. Which of course leaves us with this incredible question. As our map of cancer's inner workings gets more and more detailed, what amazing new tools are we going to be able to build to fight it? Thanks for joining me.